Emotional stress is an all too common part of our lives. We even have our own sayings such as, "I'm stressed out," or "this job stresses me out," or "relax." And we have various ways of trying to cope with it, from escaping the house to take a walk, going on vacation, or having a drink, or 2, or 3. We know moments of stress are normal, and chronic stress is bad. I'm sure you can remember a high-stress moment or period in your life where you got sick during or following the stressor. Some people will even die from these high-stress events. But what is the stress actually doing to us? Well, as the microbiome expert, I'll address its effects on the microbiome, which in turn has a large role in our immune system, and by extension, our overall health. And that microbiome/immune system combination can, for example, go on to cause autoimmune diseases. In this paper here, the authors state that for autoimmune disease, many retrospective studies found that a high proportion, up to 80%, of patients report uncommon emotional stress before disease onset. Perhaps you or someone you know went on to develop an autoimmune disease following a stressful event or a stressful period of time. For more specifics on the connection between the microbiome and a variety of autoimmune diseases, feel free to watch my 2-part video presentation on this topic. And as if this ever-present stress wasn't that enough, it can even impact us before we're even born. Research has shown prenatal stress predisposes infants and children to premature birth, low birth weight, eczema, asthma, impulsivity, anxiety, ADHD, and reduced cognitive development. Here we'll look at prenatal stress as it relates to the infant microbiome. These Dutch researchers showed that prenatal stress, as measured by a variety of surveys and to include salivary cortisol, was strongly and persistently associated with the infant's microbiota composition. When they measured 9 different infant fecal samples spread over the first 110 days of life, they found that a combination of reported high maternal stress and cortisol was related to significantly higher relative abundances of bacteria within Proteobacteria, which is a phylum full of opportunistic pathogens. High stress was also significantly related to lower relative abundances of good infant bacteria. This is illustrated by the figure to the right. The low-stress group, A, has more potential beneficial bacteria like Bifidobacterium in green, and fewer taxa, which could be opportunistic pathogens, in red. You can see how different Group B is, the high-stress group. Additionally, in the study, this imbalance was correlated to more infant gastrointestinal symptoms and allergic reactions. The prenatal stress that mom endured was transferred to the baby in the form of an altered gut microbiome, and she's the source of the baby's microbiome. We can also look at stressors in the short term, in this case, the very short term. Now, this is an experiment to help understand the connection between emotional stress, the microbiome, and disease. It's not to say that one single event like this will be the cause of your Hashimoto's disease. In any event, I'll try to explain this study without having any stressful flashbacks myself. Here, the goal in the study was to get couples arguing over marital hot topics. During these visits, a number of blood markers were taken. The researchers found that those participants with the more hostile marital interactions had higher LBP than those who were less hostile. What is LBP? I highlight it once in a while, so let's clarify it again. It's an acronym for LPS binding protein. LPS I mention all the time. LPS is prominent on the surface of gram-negative bacteria. LPS in the serum is an indicator of several things, in this case, a surrogate for gut permeability. In other words, whole or parts of bacteria, as measured indirectly by LBP, have escaped the gut and gotten into circulation. The LBP is the body's response to this escape. The job of it is to bind and present LPS to additional components of the immune system for targeting and elimination. So what this study is demonstrating is that for those who fought most aggressively over marital hot topics, they produced the most stress hormones. which then went on to do a number of things, one of which was to increase gut permeability, releasing LPS into circulation, which is something I always talk about as a bad thing. And the higher the LBP, the higher the CRP, C-reactive protein, which itself is a nonspecific marker for inflammation. So imagine this conflict not just occurring once, but many times over the course of weeks and months. Then you begin to understand the connection. In fact, something that I found powerful was that they referenced a meta-analysis which reported that the relationships between marital quality and clinical health endpoints had statistical effect sizes similar in magnitude to the health effects of diet and exercise. And for our last trial, as there are so few of these in humans for ethical reasons, we have a public speaking paper. Here they measured gut permeability, but this time with the lactulose mannitol urinary excretion test. No one test is perfect for measuring permeability. Each can be questioned, but when you look at the balance of the data in total, the individual variability within trials smooths out. Now get this, not only was this a public speaking study, but it also had a separate electric shock condition. As you can see in Figure 2, an NSAID, which is known to cause permeability, and public speech, both significantly increased gut permeability in these subjects, while shock didn't. Salivary cortisol was only increased after public speech, and the effect of public speech on permeability was only present in subjects with a significant elevation of cortisol. In other words, those most stressed out. Now understand that in this and the previous paper, gut permeability was mediated by the immune system triggered by the stress response, except for the insects. And this was proven in this paper with the use of mast cell stabilizers. Other than this instance, when I talk about permeability in my presentations, it's triggered by gut dysbiosis. That being said, now let's look at how stress impacts the microbiome and ultimately your health. First, we need to put things into perspective. What's going on in the gut is the same story that goes on everywhere among living organisms. The goal of any living thing is to reproduce. Without reproduction, a tree, a fish, a human, and yes, a bacterium cannot propagate itself. What good is anything if you're only going to be around for one generation? In order to be able to propagate, you need to have adequate resources. For example, if you're familiar with the sirtuin system, it's an ancient system of recognizing nutrient availability. And that started with bacteria and fungi and continues into our cells, which is where the resveratrol craze comes from. This system is a feast or famine switch. In famine, the body needs to change its expression to survive the day. For example, if a woman is not taking in enough carbon inputs, enough food, her body automatically knows that she will be unable to support a fetus, and she'll become amenorrheic. She'll skip her period until she starts eating adequate quantities of food again. Her body changes its expression in various ways as inputs, or lack thereof, come in and are interpreted. Whether it's the level of oxygen, proteins, sunlight, or what have you, our cells adapt to signals. I mention signals often. On the flip side, in times of feast, the body can become robust and reproduce. What's good for us is also good for bacteria. They may seem very simplistic, but they have a myriad of ways to monitor their surroundings, which can result in the changes of expression of 100 or more genes. It's about competition for resources, and every inch counts. With access to those resources under a set of conditions which favor one microbe versus the other, a bacterium can thrive and propagate. And two very important ways that bad-acting bacteria can take advantage of opportunities is via the same set of molecules, the catecholamines. The catecholamines are dopamine, norepinephrine, and epinephrine, aka adrenaline. They can act both as hormones or neurotransmitters. Although seemingly ordinary events such as high-calorie diets, a sedentary lifestyle, obesity, and hyperinsulinemia can lead to an increase in the concentration of norepinephrine, we tend to think of them in respect to fight or flight, and any traumatic injury which may follow. This makes sense, as if, for example, we're confronted with a lion, then we need instant energy to either fight it or flee. That energy comes from the rapid liberation of fat and sugar stores to power our muscles and the blink of an eye. This whole fight or flight scenario is, in theory, a once-in-a-while event. But the stress in our daily lives is constant. Does sitting in traffic with people driving like assholes piss you off? There's your lion. You arrive at work and maybe the same stupid things happen to you that occur almost every single day. There's your lion. You get home and open the mail and discover that the town is going to increase the property taxes on your home. There's your lion. The insurance company is going to increase the rates on your car 20% because of claims not related to you. Again, your lion. Or maybe you fight with your supposed loving and caring partner. Again, your lion, etc. These are all emotional events, but bring about the same end result. And on a daily, weekly, and monthly basis, there are consequences. And these catecholamines have a very interesting couple of ways of aiding bad bacteria. But first, let's go off on a catecholamine tangent for a moment. Methylation is and has been a popular topic for a number of years now. All sorts of so-called experts are throwing methyl groups at people because of the talk about the MTHFR gene. It's a fix-all-for-everyone solution. Well, not so fast. As usual, I give you information you'll find nowhere else. On some level, we're all individuals and don't all respond the same way. When you administer methyl donors, 5-MTHF, methylcobalamin, phosphatidylcholine, SAMe, or TMG, you're not just impacting the homocysteine and thionine cycles. There are many methylation reactions in the body, some of which are in catecholamine metabolism. As we can see here in Figure 1, the catecholamines start with the amino acid tyrosine, which gets converted to L-DOPA. That's the big Parkinson's drug. Then to dopamine, which is your reward pathway. Then to norepinephrine, and then to epinephrine, via a SAMe-dependent methylation reaction, a methyltransferase to be specific. So to make adrenaline, you need methyl groups. For the catabolism of epinephrine and norepinephrine, there are 3 more methyltransferase enzymes at play. Why do I mention this? Because for a number of people, and the number is not tiny, when they are administered methyl groups, they have problems with being amped up, problems sleeping, even appear to have panic attacks out of the blue. No one knows exactly what's going on, but my suspicion is with so many methyl groups floating around, there is an unregulated production of epinephrine. If you've had this experience, make a comment here on YouTube and be heard. And so again, seldom is there a one-size-fits-all solution. Now let's get back to the microbiome component of this equation. As I said, the catecholamines have a very interesting couple of ways of aiding bad bacteria. So when you get upset, the primary source of epinephrine, adrenaline, is the adrenal medulla, whereas norepinephrine is synthesized in the adrenal medulla and by adrenergic neurons distributed throughout the body. This results in spillover to include in the gut. In fact, a number of in vitro studies show many-fold increases in pathogens like E. coli, salmonella, and others when cultured with catecholamines as compared to controls. In this Table 1, I show you a wide range of references demonstrating just that. In addition, it's not just growth which is stimulated, but also their virulence, in other words, how pathogenic they become, like increasing the expression of genes involved in adhesion to the cells that line our gut. Beyond this, the table lists periodontal pathogens as well, simply to highlight for you that this is all not just restricted to the gut. In fact, when you start looking at these tables from various papers, like this one, and this one, and this one, granted they share many of the same references, but you can begin to notice the increases are for only pathogens. Whether these pathogens are gram-negative or gram-positive, the catecholamines are increasing their abundance and pathogenicity, and not the abundance and the health-promoting benefits of the good bacteria. Now, is this because the research is focused only on the catecholamine pathogen connection? Or is it because the bacteria which respond to and take advantage of stressful events are only pathogens? The latter makes more sense, as nobody ever says, I was really stressed out last month, but I got healthier from being so. Or I feel fantastic after being stressed out. And oh, by the way, my cancer and my autoimmune disease went away while I was stressed. Another of these pathogenicity traits that increasingly gets expressed is biofilm formation. Some of you may be familiar with this term. Along with adhesion, motility, and infectivity, biofilm formation is highly linked to virulence in pathogens. With that in mind, these researchers analyzed biofilm formation, among other virulence factors, and neuropathogenic strains of E. coli and S. aureus, as up to 40% of women and up to 12% of men will experience at least one episode of a UTI during their lifetime. And as shown in Figure 3, there were large increases in E. coli biofilm formation with exposure to norepinephrine. And in fact, in this study, it was shown that 75% of uropathogenic strains of E. coli and 92% of strains of S. aureus had the ability to produce biofilms. In other words, up to 80% of all strains tested. This biofilm forming ability explains how UTIs can be chronic, as biofilm formation is one of the most frequent causes of chronic infection, the formation of nosocomial strains and antibiotic resistance. And it is most often found in what I cite as the worst family in the microbiome, the family Enterobacteriaceae, of which E. coli is the poster child. And speaking of E. coli, curiously in this study, norepinephrine did not have an effect on the non-biofilm forming strains of E. coli. As I mentioned, there are two key catecholamine-driven events favoring pathogens. One is that of iron liberation. Iron is a key component of life, and especially so for a pathogen who wants to increase its abundance. Under normal conditions, the healthy body works very hard to sequester or keep locked away iron stores from hungry pathogens, and it does a pretty good job of doing it. Iron is found in a number of locations, but we'll focus on the two most relevant ones for the purposes of this conversation: transferrin and lactoferrin. This whole relationship is well established. It's simply a known fact that pathogenic bacteria crave iron for growth and virulence, and the body works hard to sequester its stores. Many studies show massive increases in pathogens with iron exposure, and these bacteria have several mechanisms to try to steal away the body's iron, but in general, they are not very successful. One of these mechanisms are via the production of what are called siderophores. Approximately 500 siderophores have been characterized, and in Figure 2, I show you two of them. They are extracellular ferrochelators, and their role is to deliver liberated iron to the bacterium. However, under normal conditions, these siderophores are not particularly effective in liberating iron on their own. But under stressful conditions, with the high production and spillover of catecholamines, the dopamine, norepinephrine, and epinephrine form complexes and inhibit the binding ability of lactoferrin and transferrin to iron, rendering them susceptible to bacterial theft by siderophores. If you thought the iron liberation story was interesting, this is perhaps even more so. The other catecholamine-driven mechanism to support pathogen growth and virulence is literally via the sensing of catecholamines on the cells of various bacteria. To date, a small family of these receptors have been identified, and you can see several of these pairs here in Figure 3. The Ne listed is norepinephrine, and the epi is epinephrine. And the AI molecule is a bacterium quorum sensing molecule, which is kind of bacteria-to-bacteria communication. So, similar sensors are able to communicate with other bacteria and with higher beings like us. These sensors and similar ones have been found in E. coli, S. enterica, and E. fecalis, all bad actors. The plugging in of catecholamines to these receptors results in the production of virulence factors. One you see listed here is for flagella, which plays a very large role in motility and virulence. Another one listed here is what's called LEE. LEE, or locus of enterocyte effacement, is simply the acronym given to the master regulator genes that are required for lesion formation. This lesion formation is well recognized, and here, in the case of E. coli, O157H7, for attaching a bacteria to colonic epithelial cells, followed by an induction of cellular structural rearrangement underneath the bacteria. This has been shown to be regulated by QSEC in several studies, to include this one here. You can see to the right of the blue arrow the increase in these pedestals with the addition of epinephrine. Another virulence factor discussed in this paper is that via this QSEC sensing, catecholamines also increase Shiga toxin production. This is a family of highly pathogenic toxins and is a defining factor for probably the most nefarious of the 5 E. coli pathotypes, the Shiga toxigenic E. coli. E. coli 0157H7 is one of these, probably the most studied. This is the kind of bacterium that hamburger recalls are made of. That's how scary this can be, and adrenaline increases its toxin production. So think about how interesting this is. Norepinephrine and epinephrine plug into a special communication sensor on bacteria and kick off upregulation of virulence factors, the ones that we know of now involved in motility, moving around, and attachment and adhesion to our cells, a bad thing, and production of a terrible toxin, a worse thing. And we know that this is the case from multiple studies like this one here. As you can see in figure E, the more norepinephrine present, the more that is bound to the key QSEC receptor, and this binding is blocked by an adrenergic antagonist. So, we know that the more stress you have, the more catecholamines you produce, the more that you can upregulate virulence factors in the bacteria in your gut. These sensors are also present in non-pathogenic E. coli, one or more species from Salmonella, and E. faecalis, and likely many more. It's fascinating to think that evolution has resulted in bacteria able to listen in on our thoughts, so to speak, and just wait for an opportune time to become opportunistic. I talk about this a lot. We always have opportunistic pathogens in our microbiome waiting to become opportunistic. Usually I discuss this in the context of food, antibiotics, PPIs, or something like that. But here it's about the impact of the thoughts in your head. You have to control the environment of your gut. If not, the opportunistic pathogens will be all too happy to control it for you. First, we highlighted the direct role that stress plays on the immune system, with the public speech study and the marriage counseling one as well. This was a direct immune system gut permeability scenario. The other two mechanisms, that of iron acquisition and microbial endocrinology, are also via stress. Whether it's chronic stress, antibiotics, PPIs, a bad diet, or all the above, these are the main drivers of the microbiome. I think you're starting to understand that the microbiome is not a static environment. In fact, far from it. All of these affect bacterial gene expression on some level. As shown in this presentation, there can be 100 or more genes turned on or off depending on iron or catecholamine availability. Many other studies show that chronic exposures to various stressors can go on to change the genetic makeup of a bacterium. I'm not just talking about expression of existing genes, but the formation of new ones, or rather what's called horizontal gene transfer, or simply by acquiring mutations. A commensal or normal-acting bacterium can evolve to become pathogenic. How is it you think a bacterium gradually becomes resistant to all known antibiotics? It didn't just start out that way. It was exposed to chronic sublethal levels. Maybe initially only 1% survived, but then that 1% serves as the progenitor for all future bacteria. This is not only the case with antibiotics, but also has been shown with antimicrobials that we produce internally. Like in Crohn's disease, there can be suboptimal levels of antimicrobials driving a more virulent version of the bacteria which survive. The point is that you need to properly treat your microbiome so it will treat you well. Not all bad bugs come from food poisoning. They are a normal part of the microbiome and can be derived from normally benign bacteria who've acquired pathogenic potential. Generally, this is a marathon we're talking about, lifelong habits which determine which direction you can take. When it comes to emotional stress It's easy to tell someone to relax, but not always easy to do it. It's up to you to modify this part of your lifestyle. Whatever measures you see fit to address the negative impacts of stress on your health. The next time you get angry, I don't expect you to think, oh, my mast cells, oh, my iron stores, or oh, my microbial endocrinology. Just realize that stress is not just in your head. There is a scientific explanation of how damaging your negative thoughts can be. How there are trillions of little critters waiting for you to get stressed out so they can take advantage of you. Maybe that thought will in some way permit you to analyze your stressors in a whole new light. If you found this presentation informative, I have many more for free in my YouTube channel and also in the Microbiome University tab on my website, themicrobiomeexpert.com. There you can select from a wide variety of topics. And if you or a loved one are struggling with a disease/condition, I have condition-specific presentations as well, along with their microbiome protocols found within its respective tab on my website.